So today, we're going to dive into this incredible story about how artificial intelligence took on one of the biggest, baddest challenges in all of biology, a puzzle that had scientists totally stumped for half a century. Let's get right into it. Okay, so just imagine this for a second. There's this massive scientific mystery, a problem so tough it's been a roadblock for 50 years. Now, what if an AI just came along and solved it? Well, that's not science fiction. That is exactly the story of AlphaFold2. So at the very heart of this whole thing is something called protein folding. You can think of it like this. Your body creates these long, floppy chains of things called amino acids. Now, for these chains to do anything useful, like, say, fight off a virus or carry oxygen in your blood, they have to fold themselves up into a very specific, very intricate 3D shape. If you get the shape right, the protein does its job. But if you get it wrong, well, that's when things can really go haywire in the body. And that right there brings us to the core of the issue, which scientists call the protein folding problem. And this wasn't just some niche academic puzzle. No, for decades, this was a fundamental roadblock. I mean, a true grand challenge for science. You know, the crazy thing is, the foundation for all this was actually laid way back in 1961. It's a really powerful idea called Anfinson's dogma. And it's simple. It basically says that the final 3D shape of a protein is completely dictated by its sequence of amino acids. So in theory, if you knew the sequence, you could predict the shape. The big problem was how. And here's where the scale of that how just gets insane. A single protein can twist and turn into an astronomical number of potential shapes. We are talking about a number like 10 to the power of 300. To put that in perspective, that number is vastly, vastly larger than the total number of atoms in the entire known universe. So trying to find the one correct shape by just trying them all out, yeah, that was completely impossible. So you might be wondering, how on earth did scientists even try to tackle this for 50 years? Well, they came up with some pretty clever methods, but every single one of them eventually just slammed into a wall. Let's take a quick look at the main approaches they were using before AI really changed the game. Okay, so on one side you had what's called homology modeling. Think of it kind of like tracing. If you had a similar protein with a shape we already knew, you could use it as a template. It was pretty fast, but if you had a brand new protein with no known relatives, yeah, you were out of luck. Then, on the other side of the coin, there was de novo modeling. This tried to figure out the structure just using the basic laws of physics. Sounds smart, right? But it was insanely complex and took a ton of computing power, so it really only worked for the tiniest, simplest proteins. So for years, the field was just inching forward, making these tiny little bits of progress. And then, boom. In 2020, DeepMind's AlphaFold2 shows up and basically causes a scientific earthquake. And the arena for this huge breakthrough? It's a competition called CASP. Seriously, you can think of it as the World Cup of protein folding. Every couple of years, the world's best teams get the sequences for proteins that have been figured out in a lab but haven't been published yet. They all submit their predictions, and then everyone sees who got closest to the real thing. It is the ultimate blind test. Okay, now this is what's so wild. Just look at the difference AlphaFold2 made at the competition, CASP 14. That score, the GDT_TS, is just a measure of accuracy. A score of 90 or more is basically considered just as good as doing it in a real lab. Before AlphaFold2, the best teams were kind of hovering around a score of 75. Then AlphaFold2 comes along and jumps all the way to 92. This wasn't just a little step up. No, this was a complete game changer, a revolution. And this visual just says it all, doesn't it? You can see the slow, steady progress over years and years of these competitions. And then you get to CASP 14 and whoosh! Look at those bars for the easy, medium, and difficult proteins. They just shoot up to a level of accuracy that nobody, and I mean nobody, thought was possible. And get this, even for the really tough proteins, the ones classified as medium and difficult that were always the biggest headache for scientists, AlphaFold2 hit an accuracy score over 85%. Before this happened, people in the field thought that kind of precision was still years, maybe even decades away. So, as you can imagine, the reaction from the scientific community was just immediate and massive. I think this quote really says it all. This wasn't just about winning a competition. People were calling it one of the most important scientific breakthroughs of our time. All right, so the billion-dollar question, how did it actually do it? What was the secret sauce inside this AI that let it crack a problem that had stumped humans for 50 years? Let's break it down. Well, a huge part of the magic was that it stopped thinking about the problem in the old ways. Instead, AlphaFold2 started treating the protein sequence like a language. It uses this super powerful AI architecture you've probably heard of, a transformer, which has something called an attention mechanism. This lets the AI look at the entire protein chain all at once and figure out the grammar, basically which parts are likely to be interacting with each other in the final 3D shape, even if they're super far apart in the simple 1D sequence. You can really boil the whole system down to three main steps. First, it takes the protein sequence and scans these huge databases to find its evolutionary cousins. That's called a multiple sequence alignment, or MSA for short. Second, all that info gets fed into the brains of the operation, a module they built called the EVA former. And finally, a structure module takes all that processed info and translates it into the actual 3D shape of the protein. And here's a look at how it all fits together. That big block in the middle, the evoformer, that's really the key. It's constantly passing information back and forth, refining its ideas about how different parts of the protein relate to each other. It's this cycle of refinement that lets the structure module build a final 3D model with such incredible accuracy. Okay, so a huge scientific breakthrough is amazing, but what does it actually mean for you and me, for the real world? Let's dig into the impact this is already having. And the answer is the impact isn't just in one little corner of science. It's everywhere. From designing new medicines to just understanding how life works at the most basic level, having these protein structures is like someone finally turning on the lights in a dark room. So let's break that down a bit. For starters, drug discovery is just being massively accelerated. Scientists can now see the 3D shape of a disease-causing protein almost instantly, letting them design better drugs faster. We're seeing people design totally new proteins from scratch to act as medicines. And we're finally starting to understand the complex dance of how all the different proteins in our cells interact with each other. It's huge. But here's the part that's just mind-blowing, the scale of this contribution. DeepMind didn't just publish their paper and call it a day. No, they used their AI to predict the structure of over 200 million proteins. That's basically every known protein on planet Earth. And then they put the entire database online, for free, for anyone to use. Now, as incredible as this all is, it's really important to remember that AlphaFold2 isn't some magic wand that solves everything. It's a massive leap forward, for sure, but we need to be clear about its limitations and what the next challenges are. See, it predicts one single static shape, but in reality, proteins are constantly moving and wiggling around. They're dynamic. It also has a tough time with parts of proteins that are naturally messy or disordered. And a really big one is that it can't show you how a protein's shape changes when a drug binds to it, or what happens when you have one tiny mutation, which is often the root cause of genetic diseases. So these are the big, exciting frontiers that researchers are tackling right now. So there you have it. AlphaFold2 cracked a 50-year-old problem, completely transforming biology as we know it. It's just a stunning example of how AI can supercharge scientific discovery. And it leaves us with this one really exciting question. Now that this grand challenge is solved, what's the next one that AI is going to help us tackle?